Now we're going to finish the amino acid biosynthesis with aromatic amino acids, which are kind of special. So phenylalanine, tyrosine, and tryptophan, they're very complex. We cannot make any of them. We can make tyrosine if we get phenylalanine, and we can add the hydroxyl group on phenylalanine to make tyrosine, in humans, I mean. But phenylalanine and tryptophan, they have to be provided in our diet, for sure. Now, as you will see, the pathway is very complex. It uses precursors both from the pentose phosphate pathway and glycolysis. From the pentose phosphate pathway to different types. We get ribose 5-phosphate from the pentose phosphate pathway that comes into the synthesis here. We get erythros 4-phosphate also from the pentose phosphate pathway. And from glycolysis, we have phosphoenol pyruvate. Actually, 2 molecules of that will come into play to make tryptophan. And from tryptophan, at some point in the tryptophan biosynthesis, the pathway bifurcates and you get phenylalanine and tyrosine. So why are they so special? They're by far the most expensive to make. Histidine comes close, which we're going to discuss next. But as you'll see, it shares several features. So for 1 mole of tryptophan, you need to spend 78 moles of ATP. It's super expensive. A little less, you could buy with a little less with phenylalanine and tyrosine. So these are really the choice pieces, let's say, of amino acids that the cell has to spend an enormous amount of energy to make them. For the organisms that do make them, there's only one way to make them. For the other amino acids, there are usually some alternatives, depending on the organism. But aromatic ones, the microorganisms that make them, and it's only microorganisms, they use the same pathway, which means it's a complex process. It was figured out once and it has been frozen in the course of evolution. Nothing has improved it. Especially in plants. Now, aromatic amino acids, it turns out in plants are super special. Not only in plants, actually, in some fungi too. In general, in organisms that make a lot of natural products, or the beautiful odorous compounds, hence the name aromatic, okay? They usually have as a precursor some of these intermediates in aromatic amino acid biosynthesis, especially tryptophan biosynthesis. In plants that tend to make a lot of these natural products and aromatic compounds, it has been estimated about 1/3 of all carbon that is produced is fixed in the Calvin cycle during and after photosynthesis. It is fluxed into the making aromatic amino acids. So animals, we cannot, all animals, I mean, from the lowly worms to us, we can only really hydroxylate phenylalanine to make tyrosine in one reaction, but that's about it. So really, we cannot make any aromatic amino acids. So all animals have to eat it in their diet. The pathway is super complex, but here is the significance, as I said, of why so many things are made out of this aromatic amino acids or intermediates of the aromatic amino acid pathway. So glycolysis and the pentose phosphate pathway are absolutely essential to give you this important intermediate called Shikimate and Chorismate. And from there on, you can make the amino acids. You can make tryptophan, you can make phenylalanine, you can make tyrosine. And here you can see what you get out of all these amino acids too. You get some neurotransmitters, you get some plant hormones like auxin. Tyrosine also you have, this is in plants what I'm showing you, but you can make dopa, which we make dopa as well. You can make other vitamins, and so on and so forth. And look at all these things from phenylalanine that the plants make, all these flavonoids, anthocyanins. These are super important, natural compounds that plants tend to accumulate a ton of. So when you are talking about aromatic amino acid biosynthesis, you don't just look at these amino acids, you see what comes from them or from intermediates during their synthesis. So the first important intermediate, it is called Shikimate. You already have the ring, you have a double bond there, and you have the hydroxyl groups. So the point of aromatic amino acids, of course, they all have an aromatic ring, right? So you can see how we kind of start to resemble something like this, not quite yet. Why Shikimate? It was first isolated from a flower, the Japanese star anise, and which has a lot of Shikimate apparently. Now, the formation, I'm showing you the next slides for a couple of reasons. So I'll tell you where to pay some special attention. As I said, the pathways are long and complicated. The pathway has many errors, as described in the Garrett and Garrett has errors in making this DAP intermediate. So here's how it's happening. First, we have these two building blocks: phosphoenol pyruvate, which we get from glycolysis, erythrose 4-phosphate, which we get from the pentose phosphate pathway, right? And those come together and to make DAP synthase. And it's this 1, 2, 3, 4, 5, 6, 7. So 3 plus 4 carbons, it gives you this 7 carbon unit. And that has to happen before it gets cyclized to eventually give you Shikimate. Okay. So this is the first step in the pathway. And as usual in these pathways, the first step has very strong product inhibition. So if you have a lot of aromatic amino acids, let's say phenylalanine and tyrosine, those will go on and inhibit DAP synthase. So you stop making them. How does it happen? These are the mechanisms, if you want to know, as opposed to reading in the book. Okay, this is the correct mechanism from the International Union of Biochemists. So the phosphoenol pyruvate will react with the carboxyl group on the enzyme, and then attack erythrose 4-phosphate, right? Which is an aldose, so it will attack this carbonyl. Very simple, straightforward reaction, nucleophilic attack, and you get DAP. Once DAP is made, then it gets cyclized, okay, to this product called 3-dehydrokinate. So the enzyme is called DHQ synthase, and that gets dehydrated further. So you have a double bond there. Okay, so after the cyclization, you have a double bond by remove this hydroxyl group. Very simple. And the last step is to reduce this keto group on the ring, and you get Shikimate. Okay, and reducing power here comes in the form, of course, an NDPH. So you start seeing the basic themes. What are those? You have intermediates, building blocks that come in from glycolysis and the pentose phosphate pathway. That's very important building blocks. And you start using your reducing power that we have talked about. That's a biosynthetic reaction. And we'll see where the ATP is coming from. By the way, phosphoenol pyruvate, it's a very high energy compound. So you see how you actually use super high energy compounds to make this aromatic amino acid, Shikimate in this case, and there's going to be more coming in. So there is the ATP, more ATP, okay, is used, ATP to phosphorylate Shikimate at the 5 positions, you make 5-phosphate Shikimate, and then the second phosphoenol pyruvate comes along and it gets attached to Shikimate 5-phosphate to make EPSP. Okay, so this is a key intermediate, as it turns out, with practical applications. If you want to know how it happens, again, that's the mechanism. But the point is that, of course, we use here the second phosphoenol pyruvate at the EPSP synthase. What are the practical implications? And before we go to the practical implications, this actually the second phosphoenol pyruvate will be removed in tryptophan, but will be the side chain. So this will become the side chain eventually of phenylalanine and tyrosine. All right. So this enzyme, EPSP synthase, that sticks the second phosphoenol pyruvate onto Shikimate 5-phosphate. We all know about it. You don't know you know about it, but you do know about it, because if you have heard of this compound, glyphosate, this compound is a super good inhibitor of EPSP synthase. And if you have not heard the name glyphosate, perhaps a more common name is Roundup. So you or your parents or your dad or whoever does yard work, I'm sure you've heard of Roundup. So you go and spray it on a plant and the plant will die, more or less in a week or two. So glyphosate is a super good inhibitor of the EPSP synthase. I should say the plant version of EPSP synthase. So you spray the plant with glyphosate, glyphosate will go on, inhibit EPSP, and will not make any aromatic amino acids. And you cannot live without aromatic precursors and amino acids, right? That's essential. So that explains why it's such a good weedicide. It kills all plants, all right? Now, it turns out that the bacterial enzyme, the bacterial EPSP synthase, is immune to Roundup. So bacteria and animals don't make, by the way, they do not make aromatic amino acids, so we do not have EPSP synthase. So we are blind to this inhibitor because we don't have the target, okay? And the organisms that do have it, such as bacteria here, they are immune to it because the inhibitor doesn't inhibit the enzyme. Now, what has happened now is to take a plant, let's say soybeans. Okay, that's a huge money maker for the big agro business here. Soybeans and other corn is the same thing, and genetically engineer them in such a way so that the genetically engineered plant is not inhibited by Roundup. Okay? And how does that happen? You take the bacterial version of EPSP synthase, you stick it in the plant. It's very conserved in evolution. Remember again that whoever can make aromatic amino acids, all these organisms, whether it's plants or microbes, there is only one way of making them. Right? That goes back to the first to the basic principle that I told you. This biosynthesis of aromatic amino acids is so complex. There is really only one way to make them. Every organism that makes them the same way. So the enzyme from bacteria can fully substitute the function of the plant enzyme. So you go into the plant that you want to engineer, you take out its native EPSP synthase, you replace it with bacterial EPSP synthase. The plant is happy now, it can make aromatic amino acids. It gets even better than that. You can spray it with Roundup, okay? And your genetically engineered plant will just grow fine, but every other plant around it, i.e. weeds, okay, it will not, because it will have its native plant EPSP synthase. And that's the whole point of this product. I believe it's sold by Monsanto, Roundup Ready soybeans. So the problem here, of course, is that then farmers go on and spray massive amounts of Roundup everywhere, okay? And in an effort to kill all the weeds, they don't care, and the soybeans grow just fine, so there's the product. This is by far, Roundup is by far one of the most commonly found chemicals, pesticides in water, even, right? And there is lots and lots of press how it may cause cancer and so on and so forth. It might. I'm sure everything in huge enough amounts will do some harm. The evidence is not entirely clear that it really does so. Okay, but who knows? If it does, it's going to be an off-target effect. Okay, because as I said, we and all other animals do not have EPSP synthase. Anyway, so special attention here on EPSP synthase. It summarizes how important aromatic biosynthesis is, aromatic amino acid biosynthesis, and some extremely applicable principles here for agriculture. So what happened so far? We started with DAP, which was again the straight up product from phosphenol pyruvate and erythrophosphate phosphate. We got to Shikimate, and from Shikimate we got to EPSP. And it turns out that all these reactions in fungi that can, if they want to, they can make, if they have to, they can make aromatic amino acids. They're all found in one enzyme. So it's a hugely, it's got all these catalytic activities. Clearly, it's an example of enzyme fusion in the course of evolution. After EPSP, Chorismate synthase will come on and clip this phosphate and make Chorismate. In the process, of course, introduce another double bond. So Chorismate is a key intermediate. You don't have to go to the aromatic amino acids like phenylalanine, tyrosine, or tryptophan, but this is a key intermediate for many natural products in plants, and let's say in fungi and plants you can make ubiquinol. Remember the coenzyme Q that we needed in the electron transport chain, or folates, which move 1 carbon units, as we discussed. They can all be made from Chorismate. Okay, so other uses for this pathway, not just for aromatic amino acids. And before we get to tryptophan now, so here's Chorismate. and this is the bottom part to get to tryptophan, you have all these reactions. And the only point that I want to make here is that a key intermediate is called anthranilate, right? Fine, but we have more input here. We have input of ribose. And a general theme that we will see here, and also in histidine biosynthesis, and also in amino acid biosynthesis, wherever ribose is used, okay, and again, ribose comes straight from the pentose phosphate pathway. That's why the pentose phosphate pathway is such an important pathway. It provides all the ribose units. It cannot be used unless it is activated first, okay, to give rise to PRPP. So there is a pyrophosphate attached to ribose at the one position. And this is the form that then gets incorporated into all kinds of reactions to generate, in this case, tryptophan or histidine, as we'll see in a minute, or nucleotides that we'll see in the next lecture. If you want to, for the organs that do make tryptophan, it is this step from chorismate to anthranilate that's specifically inhibited by tryptophan. Okay, so if you have too much of that, you have product inhibition will go back onto the first enzyme specific for tryptophan and inhibit it. So because you absolutely have to make anthranilate before you can make tryptophan. How does it work? First, at some point we're going to lose the second phosphoenol pyruvate during tryptophan biosynthesis. So first you aminate, okay, the ring here. So there is an ammonia group that's added, and then in the second step, what was phosphoenol pyruvate comes off as pyruvate to generate anthranilate. Right? So that's why we lose the second phosphoenol pyruvate. And this I mentioned already. Where this is coming from, it's coming from ribose. The ribose input in the next step, you combine ribose with anthranilate to make these compounds and go on, and eventually to give you L-tryptophan. This comes from the pentose phosphate pathway, and the reaction again with PRPP synthetase is a very important one. It adds this pyrophosphate onto, so the gamma and beta phosphates of ATP come off and they get attached to the one position here to the one position of the ribose 5-phosphate. So it's already phosphorylated at the 5 position. We're going to add a pyrophosphate at the 1 position, and PRPP synthetase does that. And that's the only way that you can activate and use ribose for biosynthetic needs. And this will happen again and again. So remember this part here. Just to remind you, ribose 5-phosphate came again from the pentose phosphate pathway. So there is one input in tryptophan biosynthesis is here, the ribose from the pentose phosphate pathway, and of course the other one that we discussed from the very first step to make DAP, it was erythrose 4-phosphate. Right? So keep building blocks, and then you go on, and we don't have to remember all these reactions. You see here you start from chorismate. As I said, you go down to tryptophan. But from chorismate, you can now go follow a separate route and make tyrosine and phenylalanine with all these reactions. Here, how important is this? There are organisms, as I said, plants and microbes that make it. So fungi makes it, for example, yeast makes it. And I work with yeast. And we hit upon the significance of this pathway a few years ago. And I will tell you some things from my research, because actually we'll get back to it when we talk at the very end, when we talk about integration. So even though organisms make it or can make it, if available on the outside in the environment, tryptophan and other aromatic amino acids, the organisms will just prefer to take it into their diet. They're not stupid, okay? Even though they can make it, they can, instead of spending 78 or whatever ATPs per mole of tryptophan, it's a lot cheaper to just take it from the outside if it exists on the outside, right? So remember that principle. So some time ago, we found that a common anti-inflammatory that I'm sure you all take, ibuprofen, Advil, okay, the brand name, actually inhibits tryptophan import. So at least in yeast, and we think in other organisms as well. Tryptophan import, and why is that important? It turns out that if you limit tryptophan uptake, you live longer if you're yeast, at least worms or flies. Right? So how do we come up with this? So here's the lifespan of yeast, measured here in generations, not in chronological time, but how many times the yeast cell can live. And we find that, if you add ibuprofen at this dose, yeast cells live longer. So that's how it extends longevity. But here's the problem. We've already talked about this in lipids, that the non-steroidal anti-inflammatories like aspirin or ibuprofen, how do they work? How do they cure our headaches? Okay, how they treat our headaches or inflammation? They block cyclooxygenases. Okay, that was an offshoot of a lipid breakdown. When you get prostaglandins, it was a slide in the lipid portion of the program. But yeast doesn't have these enzymes. As far as we know, it doesn't get headaches, right? So how does ibuprofen work? Well, these are the enzymes of tryptophan biosynthesis. So yeast has these enzymes. It can make tryptophan, right? But if it doesn't have them, it can survive as long as you give it tryptophan on the outside, unless ibuprofen is there, and then it cannot survive. That is indicated by the yellow here. The blue is other drugs that have no effect on these mutants of yeast. Okay, so you go on and you mutate each one of these genes that are involved in tryptophan biosynthesis, and in every case, that sensitizes the cells to ibuprofen, which explains this phenomenon. All right, so there are two ways, as I said, for an organism that's supposed to be yeast to satisfy its tryptophan needs. One is to take it from the outside. That's how we satisfy our tryptophan needs. That's our only way. And the other one, if you can do it, is to make it. Now, in these mutants, okay, that they cannot make it, they rely exclusively on tryptophan uptake, like we do. And it is in this setting that ibuprofen kills yeast. Okay? So in a normal setting, if they have both and you add tryptophan, all you're going to do is just going to limit the uptake. So in fact, you're going to get less tryptophan inside the cell. And we think it is that action that actually makes the yeast cells to live longer. It turns out worms also live longer if you give them tryptophan, and flies, at least female flies, live longer if you give them ibuprofen. So in all these organisms, from simple yeast to animals, okay, they take this drug, and it inhibits their uptake of tryptophan. It's as if they're on a diet, a low tryptophan diet, in effect, and that has beneficial effects, right? So if you inhibit tryptophan uptake and you are one of these species, you will actually live longer. And I will leave you with that, but we're going to get back to aging and longevity in a couple of lectures. All right. Now,